In this module, I will discuss about major scientific strategies of Human Genome Project. Human Genome Project. Human Genome Project was officially inaugurated in 1990 to map human nuclear genome. In order to coordinate different national efforts, Human Genome Organization or HUGO was established. It was biology's one of the big projects with a projected time span of 15 years, that is 990 to 2005, and an estimated cost was $3 billion. Sequencing and mapping was completed by 2003. Major objectives of Human Genome Project: to map all of the human genes, to construct a detailed physical map of entire human genome, that is 3 into 10 to the power 9 nucleotide pairs, and to determine the nucleotide sequences of all 24 human chromosomes by the year 2005. Secondary objectives. Secondary objectives were development of appropriate technologies and tools, that is DNA sequencing, then database design, etc. The first prokaryotic genome mapping to be completed was of Haemophilus influenzae in 1995. Its genome size was 1.83 megabase pair. Genome projects for model organisms such as E. coli, S. cerevisiae, C. elegans, Drosophila melanogaster, and a plant, Arabidopsis, thaliana, were also carried out. Another important objective was to work on ethical, legal, and societal implication of the project. How genome was mapped? Human genome mapping was carried out by physical methods as well as by genetic methods. Physical methods was done by large-scale DNA sequencing. On the other hand, genetic methods was carried out by disease gene localization by linkage. So, combining both the methods, physical methods and genetic methods, human genome mapping was completed. This is a figure of large-scale DNA sequencing carried out in laboratories. Strategy of human genome mapping, that is the physical methods. Human genome mapping starts with the isolation of cDNA clones and genomic clones. From the cDNA clones or complementary DNA clones, express sequence tagged EST clones and full-length clones are derived. Similarly, from genomic clones, large insert clones and STS, that is sequence tagged sites, are derived. STS are of two types, non-polymorphic and polymorphic. Now, EST, express sequence tag, then full length clones, large inserts, and STS, are mapped to the subchromosomal region by fluorescence in situ hybridization or fish technique, also by somatic cell hybrids, radiation hybrids, etc. Now, physical map was constructed by contig assembly, that is assembly of adjoining or contiguous fragments of DNA, that is overlapping segments are arranged side by side, by which physical maps were constructed. From physical map, human genome map was constructed, gene identification was made, and sequencing was carried out. Sequencing is also done directly from expressed sequence tag EST, that is the gene which are expressed. So, this is the strategy or the physical methods for human genome mapping. Next come, that is the genetic methods of human genome mapping. From families, that is disease genes are localized by linkage mapping, and disease genes are also localized by chromosomal abnormalities and also by studying the inheritance pattern of these genes. So, disease gene is ultimately localized by following these genetic methods. From this disease gene localization, genetic map was constructed. From genetic map, human genome mapping was done. From human genome map, gene identification was carried out and large scale sequencing was carried out for mapping purpose. So, both this method combining together, that is physical methods and genetic methods, human genome mapping was completed. In this figure, the pie chart showing homology of predicted human proteins to proteins of other species. Here in this pie chart, we have shown how the human proteins are related or similar with animals and other eukaryotes, with the vertebrates and animals, eukaryotes and prokaryotes. From here, from this mapping, we can conclude that the sequence of human genome revealed that there are only about 25,000 to 30,000 genes in human genome. Rest are repeated sequences, and about 60% of the predicted human proteins have similarities with proteins of other species, and over 40% of predicted human proteins have similarities with Drosophila and C. elegans. Major applications of human genome projects are in the field of medical research, for the identification of the disease gene, for prenatal diagnosis, and also in future it will be helpful for gene therapy. Besides medical research, Human Genome Project has plenty of application in forensic analysis, that is profiling of human beings. Moreover, it has got applications in anthropological research. For Human Genome Projects, its first director was James Watson, and he has made a major contribution in initiating this project. Francis Collins, he was also the next director of Human Genome Project, and Craig Venter is he has also made an important contribution in the Human Genome projects, and also the founder of cellular genomics. So Craig Venter, James Watson, Francis Collins, all of them made possible the completion of Human Genome Project. In this module, we have provided key concepts of the module, list of books for further reading, quizzes, study materials and glossary. Thank you for attending our online course.